out to the garden and then we'll kind of gather for a minute for instructions, and then you guys will do your hunt. And so when you're hunting out there, you know, one, think like a slug or snail, and two, observe, look for damage. Look for damage and see if you can figure out what damaged it, because you know it's not just slugs and snails that damage things. So, you know, look at the plants and see if you see any signs of any kind of damage. You know, and that's why IPM is so great for school garden projects, is there's a lot of different things that eat plants. So it's a cool component to throw in and brings in a lot of biology and that kind of thing, and it's pretty fun. Great, there you got all kinds of places we can look. Okay, one thing to know about this parasite, it likes wet. It'll live in wet conditions outside of the host for a while, but it does not like dry. And so I have a really good friend that grows beautiful kale, and she's like, she does the boxes, the community boxes, and she said, I tell my people, you know, about it, and I look at my kale, and if it has 3 little holes, I keep it, but if it has 5, I throw it out, and I'm like, don't do that. Dry it, you know, and make kale chips or something like that. You know, after you've washed it, you know, make kale chips.
So if the parasite die, can I assume then that the parasite is dead in there?
That's a question that I couldn't say yes or no, because the science has not been done. You know, and slime is interesting because it's got, you know, there's 4 different types at least of slime that a slug will generate. You know, it has the slime that it generates to travel, it has a slime that it generates to keep itself wet, it has a different slime that it uses in reproduction. And so we don't really know the properties of slime well enough, but I would think that, you know, that would probably not be a very likely source of getting rat lungworm from. A lot of times people would say, well, you know, I just smashed, we were having a lot of people saying, well, we're just smashing them on the ground. And we would say that's not getting rid of rat lungworm, because other slugs and snails will eat dead slugs and snails. And also, if it's wet out, those parasites are still alive. So like I said, a lot of unanswered questions. And the slime one is a really tricky one. It's a really tricky one. Because
I think that copper thing that goes around, does that work?
It works to some extent, but it's not 100%. They will cross it. And you've got to have such a wide barrier, and copper is expensive. Yeah. So to go through and copper everything is really not something that's really feasible. But people are coming up, and this is a thing, you know, like one of my schools, they made a little test box out of wood. And they would take a slug or snail and put it in the middle, and they try different things. Eggshells, you know, ashes, you know, will they cross it? And they put it in the sun where the slug's like, I want to get out of here. And, you know, and they would watch them cross this and cross that and cross this. So then one of the maintenance guys is like, well, let's try electric. And so he took a smoke detector and took it apart, and they put, you know, little wires around it, and they put the slugs in there. And they would like hit it, and they'd like fall off or slime out. And then one went up and turned sideways and didn't hit both at once. So they're like, oh, we have to move the wires closer together, you know. And so this is where, like, Lauren Pang over on Maui, the epidemiologist for Maui, he's working with kids in both Hana and Haiku, looking at using, in China, they use an electric fence to keep snails in. They're raising snails for food. So they don't want your snails leaving in the middle of the night, you know. So they use this electric fencing. And so he's trying that now on Maui. Yeah, it's great, you know. So that's the wonderful thing about this is, you know, we're doing science with the kids all the time, you know. We're science fairs and this and that. It's like, this is great. I believe the kids are going to solve this problem for Hawaii. I really do. I believe it's going to come out of the kids, you know. Scott Enright threw his hands up. Oh, we're going to lose 20 to 30% of farmers. It's like, that's not acceptable to me. That's not acceptable to me. You guys haven't tried. So let's get the kids on this, because they're going to try harder than you. And they're going to have fun doing it, and they're going to be able to, you know, meet the academic achievements that they're supposed to meet. So why not? It's fun. So anyway, what we're going to do is, you're all gloved up. Think like a slug or a snail. and it's summertime, right? So summer is not necessarily your big slug snail season. So it could be harder to find them. It might be impossible to find them. If we don't find any, we'll go back inside and we'll look at pictures of them. But what we want to do is basically just, if you do find something, turn your bag inside out, you know, and grab it and put it in there and then zip it up. And then we're going to take them back and we'll put them in the shade, you know, on one of the benches over here. And then after that, we'll look at them, we'll talk about, you know, some of the ways that malacologists use to identify and some of the different biological features of them and what they use to be able to say this is a this or this is a that. Okay? So I guess take a bag with you. If you find something, then bring it back. Make sure it's zipped up tight. They will try to get out. And then go back and look some more. This is called the New Guinea flatworm is its common name. And it's interesting that you're seeing quite a few of these here. And so what I think what we'll maybe go do back up in the lab is these guys tissue squash really well. They're very soft-bodied. To try to do a tissue squash on this is much harder because you actually have to be able to smash that tissue down to a really, really thin film. So the semi-slug and these guys work really well. And I pulled 3 of these out of the garden at Kua'okala and all of them were infected. Just 2 glass slides. Yeah. And then just a compound scope, and we could look.
And you're just looking for movement inside?
No, I'm looking for the coiled up parasites. So that one slide I had of the different stages, that one where they're all coiled, that was taken from a semi-slug that I did a tissue smash on. But I've done the same thing. And sometimes you'll actually see the worms moving around as well. So, you know, and that's what I call like a quick and dirty method of screening. You know, so running a PCR is going to take several days. You've got to, you know, you've got to excise the tissue, you've got to extract the DNA, and then you've got to run the PCR. So we've had people bring in a salad and say, I bought this at the store and there's a slug in it, you know, and they're freaked out, and rightly so. And so I'll just take a piece and do a tissue smash. And if I see it's loaded, it's like, go get dewormer right now, you know, but normally I have not. In fact, I've never yet found one that's come into our lab that I've found parasites in. So this is a good time to break into some identification things. So in looking at them, the way that malacologists look at slugs and snails is one thing is they have a mantle that covers either part or all of the body. In the Cuban slug, it covers the entire body. In the semi-slug, it covers about 3/4 of the body. So this is something malacologists will look. How much of the body is the mantle covering? When and Cuban slugs are interesting because it's like, well, which is the head and which is the tail, you know, because it's hard to tell. Underneath is the foot. So down the center of the body here is the foot, and that will produce a type of slime that allows them to stick to surfaces but to glide. And you can see the wave-like motion that they use, you know, to move along here. The antenna or the tentacles, it has 2 sets of tentacles. It has the optic tentacles, which actually has an eye. It's sensitive to light. They don't actually see things, but they're light and dark sensitive. And then underneath, below, it has little tentacles that it uses to move things to its mouth and to smell and sense things. If you know which way is the head, it has a little opening which it breathes through called the pneumostome. P-N-E-U-S-T-O-M-E. And it's not always open. And so sometimes you have to kind of wait for them to open, but they will also, a malacologist will also look at where is the pneumostome in relationship to the front or back of the slug. because it will be in different locations depending on the different species. So these are all things that are in your book. The African snail is interesting because the mantle actually comes down so far that it uses part of the mantle as its foot to kind of travel. So you can see how different the underside is. Now this guy is in the Veronichellidae family. This has been a known carrier for sure. A young woman in Maui got very infected by one of these in a salad that she paid $16 for. Yeah, it was really horrible. It has a family member. This one is called Veronichellidae cubensis, the Cuban slug. It came from Cuba. There's one that's black, that's really black and has a stripe down it. Has you guys seen that?
Yes.
That one is called Laeviculus alte, and that is a very, very good carrier. If we can find semi-slugs, we'll actually look for those. Those are real good carriers. Even way back in the 50s when they were doing, 60s when they were doing studies here, they were coming up very, very.
Is their underside whitish?
Yes, their underside is whitish, and there's a picture of them in the book too.
Yeah.
So they can come in a number of different colors, and even a lot of times the Laeviculus alte, it's like, well, in reality you would have to actually do a dissection to look at the reproductive organs to say yes or no. But
How often do they reproduce? And at what
that's
speed?
another really good thing. Those are things that, you know, we really don't know. How long do they live? How often do they reproduce? So these are great things, again, where kids are looking, the eggs, when these lay eggs on the ground, and it will have a little net-like looking structure over it. And often you'll see the slug itself curled around its egg hatch. The semi-slugs won't lay eggs on the ground. They like to attach, semi-slugs really like smooth plastic things. They really like that. So my favorite trap actually for semi-slugs is a really heavy-duty garbage bag, and I slit it open so it's easy to look all the way around. And I'll fold it, and if it's dry out, I'll sprinkle a little water on it, fold it, and then I'll just put it somewhere in the shade with a rock on it or something to keep it from blowing away. And that's our trap at our lab. We've got them under all the porches at our lab. So our traps expensive? No, they're cheap. When we ran our study and we ran all the statistics on all of our capture rates, so the kids were out there, what species is it, what trap was it under, how many, that kind of thing. Cardboard was our most effective trap. Cardboard's nice because it will stay wet. So when you're making your shelters, you really want to think about making the best hotel for these guys. So we clear the ground underneath, you know, because it's better to have dirt rather than grass, and then try to find a shady spot if we can. And actually what we found that the Cuban slugs really liked at one school was they had wood chips down between their rows, and so we put our shelters down on top of that, and between the cardboard's ability to retain moisture and the wood chip's ability to retain moisture, that was a really great trap. So snails tend to like the raised wooden board. That was our most expensive trap, was the raised wooden board trap.
How high is it raised?
It's just a 2x2 underneath it. It's a 2x2 with some boards or, you know, any old wood, you know, or you could put it on rocks. You know,
Like a plastic lid like that, or?
it's got to be high enough that their shell can fit under, you know, so they like to go underneath. So for the European garden snail, unlike, oops, sorry, something like this might work, but the European garden, they all have different likes and dislikes, you know. So would they like plastic? I don't know. You know, they might. The European garden snail, they had a lot of the big tubs that they were growing in, and underneath the lip of it, they would line up underneath there. So it's really kind of like learning what your what your species behavior is, and so that's like part of the fun of it, is the kids get to figure out who likes what, you know, and then whatever species you tend to have more of, make more traps that those ones like. You know, the highest count that we had, and all of our traps were 2 foot by 2 foot. Everything that we tested in the schools were all the same size. The highest count was 52 individuals under one trap. And it was a whole assortment of things. We had a whole assortment. We had flatworms, we had snails, we had slugs.
Like a week? Was that for a week?
It was about a week or 2 weeks, yeah. And they follow each other's slime trails. So once they start using a trap, they will follow each other's slime trails to it. And so that's, you know, you might not have a lot of success at first, but the longer you leave it out there, the more of them are going to come under. In California, we have the little Duroceras levi, which is the gray garden slug, a little guy. They have it up in Waimea too. And I have a ceramic flower pot that doesn't have the hole in it. It's like a little decorative pot somebody gave me, and I turn that upside down. Boy, they climb up in there. They don't want to lose moisture. You know, that's what they're looking at. So a lot of times you'll see a whole bunch of Cuban slugs all curled around each other, or a bunch of African snails all clumped together. It's because they're trying to retain the moisture. So that's what you're looking at. Every one of these guys is going to have a different habitat that they like. The African snails, my sister had a papaya, she's a farmer, and she growed papaya. Well, she took one of the papaya logs and split it down the middle, and so it's kind of like a gutter really, and she turned it upside down so it was like a little house. Man, the African snails loved that. They loved that. African snails really, really like papaya. And so there's a bait being made right now at Oregon University in Corvallis, and they're using papaya as the attractant. So the giant African snail is really, really distinctive. A lot of our native snails are about this size, and they're beautifully banded. They're really, really gorgeous creatures. These guys can live 7 to 8 years. They can lay a lot of eggs at a time. In Florida, when they got these, they spent $1 million to eradicate them. That's how bad of an ag pest this is, and that's how serious Florida got. They had a snail team. If you saw one of these at your house, they'd send a team out to go look for them. Yeah, I wish we'd do that here. So, again, this is, you know, a great way to be able to look at species richness and species diversity, so you can run those kinds of things. You know, that's could be more of a high school level, but with your 7th and 8th graders, a lot of the lessons, you know, as you look through them, you might see some that fit better for the 5th, 6th grade and some that are better for the 7th, 8th grade. I really like, if you've got multiple grades at your schools and multiple teachers wanting to do this, I like scaffolding. You know, if the kids in the 5th grade collect the data in the garden and then they go and hand it off to the 7th or 8th graders, and those guys put it into Excel spreadsheets and they start running the data stats and analysis and that kind of thing on it. So it's really fun. And then too, you know, we'll talk this afternoon about using ArcGIS online for reporting, but taking pictures. You know, when we were running our pilot study, I would hand one kid the camera, and they're out there taking pictures at the same time, because, you know, that's your documentation as well, and it's, you know, it's great for your story map. So, you know, have a camera, have a kid bring a phone or something like that. So basically we found 3 species here. And where did you find them? Who found what where?
I found them in the pot here, a couple of the African snails.
Were they just on the side of the pot?
They were under the plants. It's nice and dark and moist, so I lifted the plants up and found 2 in there.
Right there, all right.
Found some of the Cuban ones in the cardboard and in the tarp that was underneath that.
Great. So tarps are really good. I mean, a tarp is a great trap. If you don't need to buy plastic garbage bags, just get a little tarp and fold it up. A lot of times people will use weed cloth, like on a big island in our gardens, you know, we know how weeds love to grow, and so they'll have weed cloth over something, pull that up and look under. So a lot of times you can use what you have in the garden that you're using for control, or, you know, we used cardboard between our pathways as a weed suppression and as a trap kind of thing. So it's really a matter of getting to know who you have in your garden, what they like, and how can you recreate that, so that you're creating a situation where the diverse species have a place to go. And this is, again, why we're all going to be learning from each other. This is why we're using the ArcGIS online, because we can share each other's stories. We need to learn from each other what works, you know, and this is how we're going to build this program to be better and better and better.
I'm sorry.
You can drop the population really drastically.
Okay.
You
If
really
you're just on
can,
it.
if you're just on it. I mean, I've seen it, and that's why I believe that. I've seen my sister and brother-in-law do it on their farm. They had a 2.5-acre farm, and they farmed their neighbors a couple of acres. And they were in Wawa, the jungle was everywhere. You know, they were surrounded by jungle. And so you've got an outside source coming in. They dropped their population so far down. But you constantly have to be monitoring, you know, you need to monitor all the time. So that's why it's important to have stuff out and that you're always looking. And if you're out in the garden working, you're gonna find stuff, you know, you always do. And so that's where, you know, you have your slug jug and you go pop it in and then, you know, you're like, okay, well, I've got this. It'd be really fun to do a night class sometime and bring the kids out. An interesting thing about slugs and snails is some of them, like the leopard slug, that's another predaceous slug. And so slugs and snails will use the gardens at different times of the day to avoid each other, you know, to avoid the predators and stuff like that. And so you'll find different, the semi-slug, I usually find that early in the evening or in the morning. These Cuban guys, a lot of times I'll see them even in the daytime sometimes crawling around. But it would be interesting to learn the way that your slugs and snails use the garden. There's so much that we don't know, you know, that's what's really great about this is we're going to learn more and more and more and be able to say, you know, how many times can these guys reproduce? How long can they live?